There you go. Well, hello, my name is Imran Clark. As Eric mentioned, I'm business development with Nona Biosciences, and my talk today is going to be unlocking the potential of targeted biotherapeutics. So who are we? A little bit about Nona Biosciences. So Nona Bio is a global technology platform company. The core of our business, the foundation, is our harbor mice technology. Our harbor mice are transgenic mice that make fully human monoclonal antibodies and heavy chain only. But we also are a fully integrated biological drug discovery and development company as well. So not just finding them, but developing them and taking them through the whole preclinical process to IND to get you into the clinic. So a little bit about our harbor mice. Our harbor mice, we've got strong patent protection. We're also clinically validated. There's several molecules that we've put into the clinic that our partners have put into the clinic. We've got 2 transgenic mice. One mouse is making fully human monoclonal IgG antibodies. So we call it the H2L2. If you are familiar with antibodies, they have 2 heavy chains and 2 light chains, right? So that's a heavy chain, light chain. The other one, and it's really cool, is our HCAb. That's heavy chain only. So the light chain has been knocked out in the mice and it makes fully human heavy chain only. And that is much smaller. It's much more versatile. It's easier to work with. There's a lot of advantages to it. Also leads into our HBIS platform and other things, which I'll talk about a little bit. We are a global technology company. So we are incorporated out of Natick, Massachusetts, which is next to Boston. We have research facilities in the Netherlands and Europe, and we also have additional research facilities in China. We will take you from antibody discovery, targeted discovery, engineering, all the cell line process development, all the CMC, all the way through to IND. So we like to call it I to I, from idea to IND. And we also have a very diversified business model, which I think would be particularly interesting to startup companies. Here, they're looking for a partner that has flexibility, is negotiable, and looking more for the partnership than just the sale. And that's what we are. So why targeted biotherapeutics, or any targeted therapeutics for that matter? So if you think, especially in oncology, which is predominantly where this is applied, as well as in autoimmune and neurodegenerative diseases, but predominantly oncology, if you just give like a chemotherapeutic or a radiation therapy generally to a patient and it's fully systemic, then not only are you going to attack the disease, but you're going to attack that patient, right? So you have a lot of off-target effects that you don't want. So of course, you'd rather target your therapy right to where you want it to go, right to where the disease is in the patient. Why use antibodies? Well, antibodies are your targeting modality, right? Antibodies are designed to bind a very specific epitope. So think of like a tumor-expressed antigen, a cell that is overexpressed in a tumor on the cell surface. Use an antibody, you can target that antigen on a tumor, and either the antibody itself will be the therapeutic, or the antibody will be the vehicle that carries a therapeutic. Like, and think of a chemotherapeutic attached to an antibody. These are called antibody drug conjugates, or ADCs is the term out there. So an antibody will take that therapeutic right to where you want it. Okay, why use fully human antibodies? Okay, again, you don't want off-target effects. You don't want your patient to have an immune response to your biotherapeutic. These are foreign bodies going into a patient, into a body, and the patient can mount an immune response against the therapeutic that you're giving them. So the closer to human you are, so when people make these therapeutics in other animals, so if they were making them in mice or making them in alpaca, they would have to humanize them, but they don't get them all the way there. So it's about minimizing any kind of an immune response to your therapeutic. So if you start out fully human, then you're going to reduce that possibility. And it's also going to increase the probability that you get through clinical trials, you get into patients, and you're not wasting all this time and money and harming patients in the process. So that's why fully human. So as I mentioned, we make the H2L2 and we make the heavy chain only. Our heavy chain only, there are some other models out there, some other companies that have put heavy chain only, fully human antibodies out there. They've come around in the last couple of years. Ours was invented over 20 years ago, so it's got more than 2 decades of optimization. So a lot of the hurdles that we ran into, other companies are already running into, and trying to work that out. We've already optimized these over 2 decades. Plus, they're infringing on our patents, and some of them are finding that out right now. We are the only ones with a fully human heavy chain only that's been clinically validated. So the other ones out there, anybody that's partnered with them, those are their guinea pigs. We have proven that this platform will get molecules into the clinic and even advance past phase 1 into phase 2 and move beyond that. Okay, a little bit about it. This is the H2L2. So there's a suite of genes out there. You've got, let's see if the pointer's going to work here. Well, you have a variable region and a constant region in antibodies. So we're really concerned about the variable region. The constant region, we can make human, we can put it into a mouse, we can swap it out, very easy to do. The variable region is where all of your diversity is and where you're going to get the binding element to your specific targets, right? And so we've selected in the mouse the optimal human genes that make the variable portion of the antibody. So it's not every single gene. There's a multiple amount of genes that make antibodies and the variable region in animals and in humans, and these are combined in different ways, and this is how you get your diversity in your antibodies. But not all of them are developable as drugs, right? Some of them might be highly hydrophobic, for instance, and then when you're in manufacturing and making mass amounts, they're going to crash out. You can't really manufacture, make a drug, or they might have some other post-translational modifications that you don't want. So you want to choose the genes that are going to result in a therapeutic molecule that's going to get you through the process. So these aren't just antibody generating mice. These are therapeutic manufacturing platforms. That's how they were designed. Same thing with the HCAb. We chose the specific genes that are the most developable. And we've gone through iterations in this mouse model. So it's not just one mouse. It's been redone and redone and redone. And it's our optimal mouse. So why use heavy chain only? The HCAbs, as I mentioned, they have a lot of advantages. So this describes our Harbor HCAb plus. So it's an HCAb, it's that molecule plus another modality, a protein, plus a payload like an ADC, like the chemotherapeutic I mentioned, plus a cell, if you've heard of CAR T technology, or plus a nanoparticle if you want to target an LNP. So that was brought up as well. So something to transduce genes into a cell. Okay, so as a cell engager plus a protein, it's making your monoclonal antibody. By using the HCAb, you don't have the heavy chains on there. Instead of something that's on the order of 120 kilodaltons, you're going down to 80 kilodaltons, or you can even use just the binding element. You don't have to pair it with anything, and you get down to 8 or 12 kilodaltons. Very small, very simple. You can plug and play. You can make really interesting molecules with multiple engagers where you're recruiting a cell, you're binding to 1 or 2 even completely different antigens on a tumor cell surface, for instance. Also in the ADC world, because these are smaller, think of solid tumors and trying to get a biotherapeutic, which is a macromolecule, into that tumor. You're not just attacking the surface, you want to get inside that tumor. So you get better tumor penetration with these smaller molecules. And again, it's easier engineering. So what's popular out there now are what are known as bispecifics and trispecifics, where it's got more than one binding element, but it can bind, say, a tumor, it can recruit like an NK killer cell or a T cell and bring the immune system to that tumor and then have your immune system attack that tumor. So multiple engagement. Again, if you're doing like a CAR T and you're recruiting immune cells to the tumor environment and you've got this linker instead of having this big giant antibody, you can now with the heavy chain use just the binding portion of it. We're calling these like microbodies or nanobodies. Because it's heavy chain only, you don't have linker effects. When you have a light chain, when you have the whole molecule, you've got these cysteine residues that are reactive that form a disulfide bond that hold the light chain and heavy chain together. When you get to these nanobodies in this small component, you don't have that cysteine up there. So you have to create a linker, and that can cause problems on your therapeutic right there. There's engineering issues. There's how it reacts in the patient. Is it really efficacious? Is it doing what you want? when you get rid of that light chain component and you go to that just small binder, that nanobody, you don't have to have that linker. You don't have to link it to the light chain component to get the binding moiety, as it were. Also, it saves on intracellular space. So if you're bringing these cells together and you want the immune cell to engage with tumor cells, and you got this big giant antibody there, you think of steric effects, this huge physical thing. It might not be as effective, where if you've got these small antibodies, which are really easy to generate from these HCAbs, you can get those cells really close together. Same thing in the nanoparticle world, LNPs. There's two ways LNPs are used. It might be packed with a gene that encodes your antibody, for instance. Okay, well, if you're doing a full antibody with light chain, heavy chain, that's 2 genes. So that's taking up a lot of space inside your LNP. So when you got heavy chain only, it's a single gene that saves on the space inside that LNP, and it also allows you to pack different genetic material that you might want to put in there for other reasons. If you can also use it to deliver that LNP in a targeted way. So on the right, now you're using the nanobodies. The nanobodies now coat the surface of that nanoparticle that's carrying another therapeutic gene, and it will target that instead of just systemically delivering this nanoparticle. Now that it's, or this LNP, it's got these nanobodies on there that will find where you want it to go and take it to, say, like a solid tumor, and deposit that LNP right there and deliver that gene therapy where you want it to go. So this just shows an example of something else interesting you can do with the light chain. If you have the heavy chain only, you can skip that step. You could just pool all the B cells together. Yeah, great, we're going. Let's go backwards. Skip the B cell sorting. Just isolate all the B cells from the immunized mice in this case, because this is how you're generating antibodies in these transgenic mice. Pull out that gene and just put it right into a lentiviral library, for instance, because each B cell now only has a single gene. You don't have to worry about pairing it with a light chain gene. And so this allows you to develop some really cool display and functional assays by putting it right into a lentiviral vector, and you can combine it with anything you want. In this case, it's a CAR screening, a functional CAR screening. So there's connected it to a CAR gene and the lentiviral vector, then transduced some Jurkat cells that have a reporter construct in there. It's a GFP that's driven by a promoter that will respond to CAR activation. You then incubate with antigen. Antigen binds to cells that are expressing that one HCAb, creates a signal, lights up the GFP. You go right to FACS and sort cells. You pull out the cells that light up with GFP, throw it right into NGS and sequence it. And so now you're pulling sequences out that have already been shown to activate CAR in this case. But you could do this for any kind of a binding assay. So you could set up these mammalian display systems with the HCAbs that you can't really do as easily if you have the light chain present. So this allows a lot of versatility just on the screening side of this. And you can skip some very labor-intensive and time-intensive steps by doing this. It's really cool what this HCAb allows. I'm going to go a little faster because we lost time. This is just some examples. So this is an HCAb that's in the clinic. It's already phase 1, phase 2. It's an interesting mode of action. It acts as a checkpoint inhibitor and also as a T regulation depletion molecule. It's got a great safety profile too. So the ADA on this, the anti-drug associated effects are below 5%. It's just, again, showing examples of how you can build these different types of molecules, especially with the HCAbs. You can put them on different scaffolds. Here's a couple of antibodies. This is a cell engager 2+1 format. So this arm engaging CD3, recruiting T cells. This side's got a double binder for Claudin 18.2, which is tumor antigen. This is first in class where it's a bispecific in a symmetrical format. So you've got dual binders as the cell engager and also as the tumor antigen engager down here. So why partner with Nona Bio? Okay, We've been validated by multiple partners that see what we're doing is quite relevant. Our science team is incredibly good at what they do. The technology platform works. It's clinically validated. So we have over 70 partners now. We've been around for a couple of years. And in that time, done over 200 projects and have more than 18 molecules into the clinic already. So we're not just an antibody discovery, but we do all the engineering. It's an eye to eye, all the CMC, all the PK, the in vivo, cell line and process development. Everything is in-house and under roof. So you have everything you need with one partner to get you from your target and discovery all the way to IND and pushing you into the clinic. Example, we also have an incredible ADC team. So those are hot right now. So part of our workflow. We've got off-the-shelf payloads and linkers, but we also have unique proprietary linkers and payloads. So we've got a variety of things for you to pick from and mix and match from. Our HCAb molecule, because the light chain's not there, exposes those two cysteine residues. And instead of mutating them out, they're reactive. You can use them to link a chemical to it, like a chemotherapeutic to it, an ADC. And it turns out those two sites are much more efficacious in sites that are typically used lower down on the hinge region in antibodies, where there's what's called a DAR, a drug to antibody ratio. So this would be a DAR of 2. When you use sites and people tried to make DAR 4s, DAR 6s, DAR 8s on the lower region, you think more would be better and more efficacious. Our DAR 2 beats the DAR 8s just because of where it's positioned. It's more stable and it's more efficacious. And this is proprietary to our HCAb. Nobody else can do this. We've got a great development group for bispecifics, trispecifics, multispecifics, especially using the HCAbs. You can really just plug and play like Legos and Lincoln Logs and build whatever molecule you want. It's much more simple to work with. We got DMPK capabilities in-house, all of the CMC capabilities. Okay, and then why, again, partner with us? Let's just talk about the flexible business models, particularly on the service side. I mean, we can license out the platform. We also have assets, molecules that we've already developed that people will license. But often people use this for the service, right? Discover a binder to their target and build this molecule for them. A typical model out there, and it encumbers, especially new startups, is that the models out there have upfront fees. They have lots of milestones through the development process, the commercial process, and royalty. We don't do that. We're just a fee for service with a single milestone upon you reaching successful IND, and that's it. So if you need to raise more capital, sell the company, you're not encumbered by us. There's no royalties, there's no commercial milestones, there's no late clinical stage milestones, just one IND, and then just paying for the actual work that's done in the fee for service. And there's no technology access fee either. So we keep it real simple and client friendly. And even that on the IND milestone is negotiable. We want to work with you. So you're like, hey, I need to get to where I'm like, like 5th patient dosing in phase 1, because that's when we're going to raise capital and find out what goes on. We can talk about that. So that's a very general overview of Nona Biosciences. I really appreciate your time and your patience for the little snafu. One in the back.
I might have missed this. What's the timeline from, say, here's the target we're interested in, to here is an antibody that binds the target and we have characterized it and it works the way we think, the way you want it to?
Great question. So it's going to depend on the workflow you go with, right? But the timeline is typically between 6 and 7 months, 8 months. Sometimes we do faster than that. Again, it depends on the workflow, but that's pretty typical. So, and it also depends on how ready your antigen is. So are we part of the process of designing the antigen and then immunizing the mice, or do you already have that ready and set to go, right? And then we're going to immunize. But from immunizing the mice and then going through our process, and now are you going to go through our HCAb and like that mammalian display? Are we going to do the single B cell sorting and beacon, which is actually very popular? Less popular is phage display, use display. We can do that. Not many people are doing that anymore. That's your old school. But yeah, figure 6 to 7 months on average. And with some characterization in it too. We're going to have like binding affinities and TMs and, basic characterization going on.
Thanks.
You're welcome.
Completely separate question, if no one else has one.
Yeah.
Does Nona plan to have an internal pipeline of their own at some point, or is it just a partnership company?
Nona itself, we're not really developing our own internal pipeline much. We do have assets that we license out. So we're like the new company that's been around a long time. It's kind of like my joke because we're a spinoff from Harbor Biomed, right? And so really we are now monetizing as a service everything that Harbor Biomed had created internally with these mice. And so we license out those assets, and those assets came about through many different ways, either proof of concept studies or they did something on purpose. So they came for several different reasons. Through Harbor Biomed, we are working on certain assets that are popular right now because they'll be easy to license. For like in-house right now that we have already is a suite of CD3 binders, including single chain only HCAbs, and even from another animal, alpaca, that's really well characterized. So we've just got a whole suite for people to look at, and that's hot right now. So yeah. But for the most part, we're service, and it's through partners. And our partners own everything. So we create a library for you, and you pull a binder out of it, and it's your favorite one, you go up and develop it. We're not going to take that library and use it for another customer. You own that for life. Right? I mean, we might do another discovery project for the same target, unless you pay us not to.
Thanks.
Yeah. Thank you so much.